Welcome to the second Front Rows lecture series at the Scripps Research. Thank you for joining us. It's really our pleasure to have you all here. My name is Kirill Marteminov. I'm a professor and chair of neuroscience here at the Scripps Research Institute, overseeing the interests in neuroscience of the institute in this area. As you may know, Scripps Research is an independent biomedical institute ranked the most influential in the world for its impact on innovation. It's also a non-for-profit charity, one given the most influential 4-star rating for financial transparency and accountability by Charity Navigator. With 2 campuses, one in La Jolla, California, and one right here in Jupiter, Florida, we're advancing human health through the discoveries in basic science and preclinical research that address pressing medical needs. Scripps Research is also the world's most renowned scientific institutions for its discoveries in brain science. Our mission is to understand basic brain mechanisms, discover what goes wrong in all sorts of brain diseases, neurological, psychiatric disorders that affect the brain, and develop new cures and therapies for them. Our areas of emphasis in particular include addiction, memory loss, brain development, and neurodegenerative conditions. And today, we're hoping to provide a glimpse on the exciting discoveries in this area with a lecture by Dr. Lasmézas on the therapies of brain diseases. First of all, I wanted to apologize that Dr. Miller, who was originally advertised to deliver this lecture, is unable to present. Dr. Lasmézas will lecture today, and let me tell you that you are in for a real treat, because you're going to hear from the brightest scientific minds working at this institute. Dr. Lasmézas is a professor at the Scripps Research. She is internationally recognized expert in the field of neurodegenerative diseases due to misfolded proteins. And those diseases include Alzheimer's, Parkinson's, and ALS, and also prion diseases. She completed her doctoral education in France and worked at the medical research branch of the French Atomic Energy Commission before being recruited to Scripps about 15 years ago. We got really lucky. Dr. Lasmézas is world famous for providing her the first experimental evidence that mad cow disease could be transmitted to humans. Her work led to the characterization of a new type of infectious agents made of proteins now known as prions. Dr. Lasmézas discovered how they travel from the digestive tract to the brain, how they hijack the immune system, and how they multiply before invading the nervous system. Dr. Lasmézas is highly sought as an advisor, serving on numerous governmental agencies and public health committees in the US and actually across the world. And this includes World Health Organization, the British Department of Environment, FDA, EPA, USDA, just to name a few. Dr. Lasmézas' current research is at the frontier of identifying key players in the pathogenic process leading to neuronal dysfunction and death, on developing novel disease-modifying treatments for Alzheimer's, Parkinson's, and prion diseases. Dr. Lasmézas is also a founder of VIT, a Jupiter-based therapeutic startup company, working to advance treatments for the Alzheimer's, Parkinson's disease towards clinical trials. So with this, it's a real pleasure to have Dr. Lasmézas deliver this talk, and please join me in welcoming her to the podium.
Thank you so much, Kirill, for this very kind and over-generous introduction. Thank you all for being here. I really appreciate my colleagues at Scripps. I appreciate your presence also very, very much. So don't be afraid of this kind of device I have in my back. Dr. Martimianov told me already I look like a ninja turtle, so it's not anything dangerous. The reason for that is that they had to put on the microphone somewhere. So this is the solution we found, the scientists, was to hang this thing on my back. Anyway, so let's get us started. So we're going to talk today about, where are we in understanding brain diseases? And that goes along with understanding brain function, of course, but specifically today I'm going to speak about brain diseases. Unfortunately, I have to. And also, more excitingly, toward what we're doing towards disease-modifying therapies. And so I would like to start by saying that my hero, right after Spider-Man, is the neuron. And why is that? Well, neurons, they're the brain cells, you know, and they are the functional units of our brains. And I don't know if you realize that our brains basically are doing everything. We can't think, we can't walk, we can't feel, we can't move, we can't love, nothing without our brains. And the little busy bees inside our brains that make that possible are these cells here that are called neurons. So I will call them brain cells or neurons, and they have different shapes. So this is just a schematic view of a neuron. And so neurons love to communicate. Actually, they need to communicate. They're like people, but people don't know that. And so if they don't communicate enough, they get sick and they lose their connections. And how do they do that? How do they communicate? So they do that by making connections with the neighboring neurons, and then when they have an electric impulse that stimulates them, they release a little molecule at one of their extremities, and this is depicted here by the little green dot that you can see here. And this little molecule called a neurotransmitter. Have you heard of serotonin, acetylcholine, and dopamine? So these are neurotransmitters. So these are these little molecules that are released and they're taken up by the next neuron here that then gets stimulated and transmits the signal to his neighbor. So we have 100 billion of these neurons. Each of them makes 10,000 connections. So I just showed one connection because otherwise you wouldn't see anything. So I think that explains my fascination for the brain, the fascination that all my colleagues at Scripps have in the Department of Neuroscience in particular for the brain, how it functions and what happens when it goes wrong. And it explains why we're so wonderful precision machines. When we make a movement, usually it's just the right movement regulated all the time. Talking, for example, is an extremely complicated task because we have to intake auditive, visual stimuli. We have to integrate them in our brain. Then we have to think about what we're going to say, the words we're going to find, and then we have to speak them out to activate the right muscles to do that. Have you ever thought about what our brain, how hard-working our brains are when we do anything? So what we do at Scripps Research here in Jupiter in the neuroscience department is we're working on how memories are formed and stored. That's important to store them. Genetic pathways involved in aging and nutrition, neurodevelopmental diseases like autism, neurodegenerative diseases, and I'm going to talk a lot about those, blindness, sensory perception, and the treatment of pain, how to better treat pain without having all the side effects that, for example, opioids have. And before I finish talking on this slide, I would like to take a little poll, if you don't mind. And so the question I'm going to ask you, and please answer something. It's pretty easy. There are only two answers, one or two. So the question is, if we had the capability, the technical capability, to determine our memory settings for life, would you choose setting number 1, which would be to remember everything, or would you choose setting number 2, which is to only remember good memories? So remember, setting number 1, everything, we remember everything. And we never forget. And setting number 2, we only remember the good memories, and that's it. So who votes for number 1? Well, okay. And who votes for number 2? Okay, so the vast majority of you voted for number 1. Which is interesting. It's very interesting to me. That was a real-life experiment. I didn't know what you guys would answer, by the way. I had no clue. And it's very interesting. And that shows you that, you know, we all have different perceptions of how we should function, and we make different choices, right? That's why it's so interesting that we're different and make different choices. But from a medical perspective, so most of you want to remember all memories. From a medical perspective, it can be a problem to remember everything. And this is what Courtney Miller, who was supposed to talk this afternoon here and couldn't because she had a little health problem, but you come back when she talks, right? Because she's going to tell you everything about the fact that some memories are associated with addiction relapse. If your brain associates a particular memory with a situation, you will have a tendency to relapse in your addiction. Other memories, and this is very well known, are traumatic memories, of course, and cause PTSD. And her lab found a mechanism that is operating in the emotional part of the brain that is responsible for establishing the memories associated with addiction relapse. So now she's developing medications to be able to remove those memories. So I hope you appreciate the importance of this work. So neurodegenerative diseases now. What are they? So Alzheimer's disease. 5.8 million people in the United States, about 40 million in the world, probably more, because we just couldn't count all of them. Costs $277 billion per year to the American society in medicine, hospitalization, time that unpaid caregiver give for the patients, and early retirement, lost work hours. That's even more for Parkinson's disease, it's even more the case. And the curve is going up like that, which means that in 2050, if we don't do anything about these diseases, there will be 15 million in the US, and it will cost more than $1 trillion to the society. So it's more than time. We have to do something about these diseases. Otherwise, it's going to be a catastrophe. So Parkinson's disease comes second with 1 million people affected. And then dementia with Lewy bodies has the same molecular mechanism as Parkinson's, and are almost as many people who have dementia with Lewy bodies as there are people having Parkinson's disease. Other diseases that you might have heard of, fronto-temporal dementia, amyotrophic lateral sclerosis, Huntington's disease, prion diseases. So prion diseases are very, very rare, and I spent quite a part of my career working on prion diseases because there are animal disease and human disease. Unfortunately, some animal diseases transmitted this disease to humans. And it's a disease with a very rapid fatal outcome. So seeing this list of diseases, I think you're not very surprised now to hear that about more than 10% of people over the age of 65 have a neurological disease. So I'm going to start by talking to you, instead of giving you a list of symptoms for Alzheimer's, I'm going to talk about the very first patient who was described having a disease that was later called Alzheimer's disease. This patient was a 51-year-old woman called August D. And she was admitted at the Hospital for Mentally Ill and Epileptic in Frankfurt in Germany because she presented the following symptoms. She lost memory. She started developing a very strong sense of jealousy towards her husband. She was disoriented, bringing objects back and forth in her apartment, and had very profound personality changes and also auditory hallucinations. There was a young doctor in this hospital, Dr. Alzheimer, who spent 4 days with her, giving very thorough interviews and taking notes of what she was responding. And I'm going to read to you a little excerpt of these interviews by Dr. Alzheimer. Alzheimer's notes in the file begin on November 26, 1901. He asked simple questions and wrote down August D's answers systematically. She sits on the bed with a helpless expression. What is your name? August. Last name? August. What is your husband's name? August, I think. Your husband. Ah, my husband. She looked as if she didn't understand the question. Are you married? To August, Mrs. D. Yes, yes, August D. At lunch, she eats cauliflower and pork. Asked what she's eating, she answers spinach. Writing. When she has to write Mrs. August D., she writes Mrs., and we must repeat the other words because she forgets them. The patient is not able to progress in writing and repeats, I have lost myself. And I think this describes the tragedy of Alzheimer's disease, because thinking is what makes us human, what makes us who we are, and if we lose the capacity to think, I think we're losing ourselves. So, what are the risk factors? There are risk factors that there is nothing we can do about. Age, gender, and genetics. Gender. By the age of 65, 1 out of 6 women will develop Alzheimer's disease during the rest of her life. Only 1 out of 11 men will develop Alzheimer's disease during the rest of his life. So there is a gender inequality here. Genetics, there are other genetic factors. I'll come back to those. So there's nothing we can do about, obviously. But there are some risk factors we can actually do something. And those are risk factors that you all know about. But we're not sure if we should take them so seriously, or we know about them, but what we don't know is that they're actually a risk factor for Alzheimer's because it's not directly intuitive, such as eating healthy. Obesity is a big risk factor for Alzheimer's disease. Having high blood pressure, being depressed and not treating your depression, not exercising enough, or not interacting socially enough. I told you the neurons love to communicate. Well, human beings need that too. And that helps the brain to establish more connections and to become more resistant to Alzheimer's disease. And then there are environmental risks, such as exposure to pesticides or even head trauma. What are the treatments? Unfortunately, there are not much treatment options. Most of the patients are giving cholinesterase inhibitors, and what these medications do is that they prevent the breakdown of one of the neurotransmitters, called acetylcholine, so it helps a little bit the neurons communicate. But that only works if you still have the neurons there to be able to communicate in the first place. So typically, it doesn't work in all the patients and for a limited period of time. Parkinson's disease. So Parkinson's disease is classified as a movement disorder. And it was described by Dr. Parkinson's now at the beginning of the 19th century in his essay of the shaking palsy. And the most common signs are tremors, rigidity, slow movements, imbalance, and falls as the disease progresses. There are also non-motor signs, cognitive. Some patients, they complain that they're thinking too slowly, and in some cases it even evolves into dementia. And other symptoms are sleep. So Parkinson's disease patients, sometimes very early on, they act out their dreams. So you know that there is this sleep phase that's called the rapid eye movement phase, where we dream, but there is a kind of inhibition so that we don't move, right? When we dream about flying, we don't try to fly, luckily. And so, but in Parkinson's disease, some patients, they act out their dreams, so it has all the consequences you can think about. And also depression. Depression is a real symptom. It's not associated because the person knows about the sickness. It's a real symptom that needs to be treated. So the risk factors are very similar to Alzheimer's disease. Age is a major risk factor, and actually I think, yes, I have a curve here where you can see it very clearly. This is, you know, the prevalence of Parkinson's disease as a function of age, and you can see here how the curve takes off after 60 years. Here, the gender, it's the opposite. Men are twice as much at risk as women are, exactly the opposite of Alzheimer's disease. And nobody really knows why. There's a lot of studies done by researchers to understand this better, the influences of hormone, neurodevelopment, etc. But we're not quite there yet to really understand what's going on. There are genetic factors, exposure to herbicides and pesticides, to the point that we in the lab, we actually use pesticides and herbicides to model Parkinson's disease in the test tube. So we have to be careful with that. The treatment. So Parkinson's stands out a little bit from the other diseases because for Parkinson's disease, there is at least pretty effective symptomatic treatment. What do they do? So in Parkinson's disease, the brain region that is affected is a very localized brain region, at least at the beginning, called the substantia nigra. And what the neurons produce here is dopamine. It's the neurotransmitter dopamine. As the neurons get sick, they produce less and less dopamine. So there is, since the 60s, a medication which we call dopamine replacement therapy that replenishes the brain for dopamine. Basically, patients take dopamine precursors or agonists. And so it helps the neuron communicate. But, and another possibility is to stimulate the neurons directly surgically by implanting electrodes in the right region of the brain, stimulating the neurons. So that works pretty good as palliative treatment for Parkinson's disease, at least during some years. But there is also a limitation. The neurons still have to be there. They still have to be able to respond to the treatment. And after, if the neurons are gone, dopamine replacement is not going to help anymore. So here again, what we call disease-modifying treatments in our scientific jargon are treatments that stop or at least slow down the progression of the disease itself. It would be a treatment that makes the neuron feel better, so they stop being sick and they heal themselves. That is a disease-modifying treatment, as opposed to palliative treatment that I described to you that only treats some symptoms, but the disease course stays the same. It doesn't change how the disease progresses. Finally, I'm going to describe rapidly amyotrophic lateral sclerosis. It's also a movement disorder. And here what happens is that the neurons that are affected are the ones that directly command the muscles. So if the neurons are sick, the muscles don't respond, they don't get stimulated anymore. And so it's characterized by a muscle weakness that starts somewhere, depending on the patient, and becomes generalized. This is the disease that has, from the three that I described, the shortest life expectancy, 2 to 5 years. Only 2 approved treatments that prolong your life by a maximum of 4 months. And risk factors we don't really know too well, but smoking, lead exposure, and military service have been incriminated. Again, unfortunately, no disease-modifying treatment. That's why we're here, working on those treatments. But why? Why don't we have treatments for these diseases when we have such wonderful treatments now for so many other diseases? So it's simply due to the fact that the mechanisms operating in these diseases is fundamentally different from the other diseases. For example, in infectious diseases, you get infected by a microorganism, a microbe, right? So it could be a bacteria, and since the availability of antibiotics at the end of World War II, that really changed and saved billions of lives. Later on, we developed antivirals that now combat viruses, another type of microorganism. We're still working on the coronaviruses, by the way. And then you can have parasites or fungi, but for all these classes of infectious agents, scientists have developed medication, and what do these medications do? They either kill the microorganism or slow down so much their multiplication that our own body gets rid of the rest. Okay, then you have cancer. What happens in cancer? Well, some cells in our bodies escape the cell cycle regulation that makes them, do their function, and then they die off, and new cells are being made. When they escape the cell cycle regulation, they start multiplying like crazy, and you can't stop them multiplying, and they form a tumor, right? And sometimes the tumor disseminates and makes other tumors in distant organs. So what are the therapies doing here? They're killing the cancer cells. And the challenge here is to have therapies that will be specific for the cancer cells and don't touch your other cells in your body to not make you sick. And it's pretty difficult to achieve, and we've become so much better at doing it in the last 10 years or so. Okay, but in each case, we're trying to kill a microorganism or kill a cell that became crazy. In neurodegenerative diseases, the neurons die off by themselves. You want to do the opposite. You want to rescue them. So this is why we don't have therapies, because this is extremely difficult to do. And for a long time, we didn't even know what is the cause, what makes them die. So this is where all the tremendous advances have been made in the last 15 years, and specifically in the last 5 to 10 years, I would say. So this is what neuropathologists see in the brains of patients who have one of these diseases. What do they see? They see these dark dots here. These are the dark dots in the brain of a Parkinson's disease patient. They're called Lewy-bodies. Maybe some of you have heard of Lewy-bodies, because there is also Lewy-body dementia, which is related. In Alzheimer's, they're called plaques or tangles, depending they're outside or inside the cell. And in Creutzfeldt-Jacob disease, they're also called plaques. Any of you has an idea what these plaques, tangles, what these dark dots are made of? I can leave. You all know already. Yes, that's right. They're made of proteins. Exactly. So proteins. So you know that we're mostly made of water, but we also have lipids, sugars, and proteins. Proteins, so originally, I mean, it all starts with the genes, our DNA, that makes RNA. RNA they have their own life, right? And Dr. Matt Disney will tell you everything about RNAs. But then some RNAs make proteins. And proteins have multiple functions in the body. They do pretty much, I mean, everything. Some proteins are there because they're structural proteins, so they keep the structure. They're the walls, basically, of our organs. And even inside the cell, they keep the structures in place. Some proteins, they transmit signals. For example, this is an antibody here it has a little part that senses microbes or cancer cells, will recognize them, and then the other part of the protein will tell the immune system, hey, there is an invader here, go kill it. So they're signaling proteins. Some proteins are enzymes, so they help reactions. Reactions, this is what happens all the time due to our metabolism when we make energy. And then some proteins are transporters, so they're called carriers, and they carry all kind of stuff like metal ions, lipids, cholesterol, elephants, etc. So as you can see, to be able to do that, they have the right shape, right? If you take a signaling molecule and ask that molecule to carry this guy, it will be a problem. If you have an enzyme that's supposed to have this pocket here to accept the ligand, and it doesn't have the pocket, it's not going to do its job. So shape is key with proteins. So now what happens in these diseases? And once you know that, you know the cause of all the diseases I've previously described. Proteins must have the right shape. When we're healthy, everything goes well. This is what happens. So imagine we're making trillions of proteins per minute. So we're making tons of proteins as we are right now. It's a very complicated process. I told you, DNA has to make RNA, and RNA makes the protein. They have to have the right shape, go to the right place in the cell. Sometimes something goes wrong. And when something goes wrong, they change shape. Luckily, we have quality control system, and we have other proteins called chaperone that patrol around looking for the bad guys. And if one has the wrong shape, so here in this depiction, the good protein is supposed to be a square for some reason. Let's say it's a structural protein, right? And it's a brick of a wall. It has to be a square. When it becomes like abnormal, it's all rounded up. So the chaperones, they will stick to it and force it back into the right shape. Sometimes that too fails. So we have a second quality control system. We have other recognition molecules that will stick to the bad protein and send it off for degradation, get rid of it, make a new one. And that works wonderfully. I told you we're really precision machines. For some reason, sometimes, the quality control system is weakened, right? Imagine like when we have a weakened immune system for a reason and we will catch an infection. So here, this quality control system can also get weakened, sometimes because we have a genetic mutation that favors that or for another reason. And when that happens, well, the misfolded protein, I call it misfolded, it has the wrong shape, persists. And when it persists, it becomes very dangerous because there are sticky surfaces on the protein that the proteins hide inside. But when they have the wrong shape, they exhibit them at their surface, and they become very sticky like flypaper. And they start forming clumps. And when that starts, it doesn't stop, it continues. And this is, Kirill told you earlier that my lab for years was working on prion diseases. Prion disease, the prion protein is one of the first proteins for which we demonstrated that this mechanism is self-amplifying. So you have, a templating process, and the aggregates, we call them aggregates, the clumps, right? They grow and grow. Sometimes the cell manages to cleave them off, but then it starts all over, and you have this vicious circle with these clumps growing bigger and forming these plaques in the brains of Alzheimer's disease patients or the Lewy bodies. So now you're going to tell me, if it's the same mechanism for all these diseases, why are there different diseases? Well, it's simply because, depending on the disease, that happens to a different protein. So in Alzheimer's disease, the proteins that get misfolded and form the clumps are amyloid beta and tau. In Parkinson's disease, it's alpha-synuclein. That's the protein that forms the clumps. In ALS, there are several of them: FUS, CDP43, SOD, etc. And the risk factors, the genetic risk factors, they don't change shape, but they have a role in the cell that ultimately, when they misfunction, either because they're involved in degradation of the bad proteins, etc., eventually it leads to the accumulation of those proteins. So then that explains to you the current focus of my laboratory. Since the proteins are the culprits, what are you going to do about it? Well, you're going to try to find which of these protein clumps are really toxic, are really the bad guys. First of all, to be able to get rid of them, right? And second, you want to understand how exactly do these protein clumps attack the brain cells? What do they do to these brain cells to make them sick? So you can also do something about it. And so for the first point, maybe I should go back real quick to this slide. So for a long time, I mean, the first neuropathologists, they saw the big clumps in the brains of the patients. So scientists thought these are really the toxic proteins, and they started developing therapies, specifically for Alzheimer's disease, directed against those big clumps. Unfortunately, and these are all the failed clinical trials that you might have heard about, it didn't work. Why? Because the big clumps are not the ones that really damage the cells. It's the smaller ones. And this has been discovered much, much later. But we still don't know exactly which ones. So I'm going to give you one example of what my lab did in each of our focus, right? What are the toxic clumps? Second, what are the mechanisms by which the neurons get sick? So this is the first example. What we discovered very recently, what we think is one of the most toxic protein clump in Parkinson's disease. So what we did is that we took brain cells and we cultured them in a Petri dish. And then we, in the lab, made the equivalent of Lewy bodies, right? We made alpha-synuclein change shape, form clumps, put on the neurons. And the neurons, and we cut them into little pieces so they're taken up a little bit easier by the neurons. The neurons take them up. And then I told you that these clumps become bigger in the neurons, remember? And the neuron is trying to get rid of them. So what you see here, this little pocket, it's a garbage bag. It's called an autophagic vacuole, but what it really is, it's a garbage bag that engulfs the big clump and then digests it. And this works very well for a while. But if there is too much of the clump, the poor neuron just can't deliver anymore. So it sticks around. And you can see that this neuron starts to be sick. It lost its nice extensions. It can't communicate really good. And then it's also if the neuron can't communicate anymore, it becomes even sicker. So it becomes even less efficient at degrading the clumps now. And things don't get better. And here is how it looks like when it's almost, done. You have huge garbage bags. And then I'm going to show you how they look like in real life, when we make pictures of the neurons. And these are the big protein clumps. So now, what you see here in blue are called mitochondria. Mitochondria are our own energy factories. We wouldn't be able to do anything if we wouldn't produce energy all the time, right? Our brain, by the way, is a very big consumer of energy. 20% of our total energy is consumed by the brain. So these little organelles here make all this energy. We knew that they're damaged in Parkinson's disease and Alzheimer's. When we say we, the scientific community as a whole knew for several years mitochondria have a problem in Parkinson's disease. But where we were scratching our heads is that the big clumps that I told you that are the equivalent of the Lewy bodies, they don't even touch the mitochondria, or sometimes they do, but not systematically. So we couldn't find a direct relationship of what was really so bad to the mitochondria. Until a talented postdoc in my laboratory found that there is something else. And this something else are the little aggregates here that you see in red. And so what happens is that the cell is trying to degrade the big Lewy-body type of aggregates, but it's overwhelmed, remember? So it releases little pieces. Instead of completely destroying it, it releases little pieces. And these are really the bad guy. Because these little clumps now, they have a high affinity for the mitochondria, and they literally sit on the mitochondria and kill them. And I'm going to show you real pictures of how it looks like when they start killing the mitochondria. But first I wanted to show you how it really looks like under the microscope. So you see here in blue, that's the DNA. So you only see what we call it labeling. We put little probes that can detect specifically what we want to see. So the blue is the DNA, and the green is the Lewy body, right? And this is 2 days after making the neurons, putting the bad Lewy bodies on the neurons. 6 days later, I told you this is a self-amplifying process. So now you can appreciate this because the amount of these green fibrils because it's like elongated, has increased. But you can see also that the little red dots have appeared. If you wait longer, 14 days, you can see that the neurons are full of these Lewy body kind of clumps, but they also have more of the toxic aggregates. So this is the equivalent of the scheme that I showed you earlier. And we published that 2 years ago in PNAS. And here is what I wanted to show you when I told you how the mitochondria become sick. So this is a scheme showing you a mitochondria that is kind of sliced in the middle. And it looks like a big blob. It's an energy factory. So it has a membrane outside, and it has an inner membrane, and here a lot of little proteins, very, very busy making energy. The energy can only be made if there is an electric gradient inside this membrane, okay? It's called the membrane potential. Okay, what here we wanted to show is that this is here a little probe we put just to show the shape of the mitochondria. So this is kind of, if you want, we're looking at the outside, the shape of the mitochondria here, this like white blobbish picture. And then we specifically detected the little aggregates that I told you are so bad, and we put a little probe that senses the presence of these clumps here. And when we superimpose the two signals and we do some pseudo-coloring to see, so this is red here, this is supposed to be blue, we put two together, you see it's pink because the aggregate sits on the mitochondria directly. It's directly on the mitochondria. And then what we did is that we put a little sensor for the electric potential. If the mitochondria is functioning, you have a signal. If it's not functional, no signal. And you can see here the big void where P-alpha-synuclein star is sitting, there is no electric potential. Because P-alpha-synuclein star, we called it P-alpha-synuclein star. Our executive vice president told me I should have chosen a more simple name, so we have to think about one. But so wherever this P-alpha-synuclein star is sitting on the mitochondria, it's slowly killing the mitochondria. It already cannot work properly. And then we have other pictures where we see that the mitochondria is cut into little pieces and it's sent for degradation. So this is what happens. So what are we going to do about it? Well, now we have to determine the structure of that bad guy. We know there's this little clump that is very toxic, but as long as we don't know what's the shape, what makes it so toxic, what is it that interacts wrongly with the cell, what are its characteristics, we won't really be able to do too much about it. And so cryoelectron microscopy is a method to determine the structure of proteins in their native hydrated form, and it can even determine the structure of pretty big proteins, and that's what we're talking about here. We're talking about these fibrils or clumps, so they're all very big, and classical methods have not been very efficient at solving the structure of those type of proteins or protein clumps. And during the last few years, tremendous advances have been made in cryo-EM, cryoelectron microscopy, to the point that it's called, some people write that it's revolutionized the structure determination of biomolecules. And 3 scientists have won the Nobel Prize in 2017 for the advances they made on cryo-EM. So here, for example, just to show you what it can do, this is the structure of F-actin, which is a protein in, we all have it, but here in this one is the one from the malaria parasite. And the structure you see, it shows the shape exactly of this protein and even the little pocket where it's situated and where the drug can fit in. So this is a 3.8 angstrom resolution. Do you know what it means, a 3.8 angstrom resolution? It's about 10 billion times smaller than an inch. So that's what it's doing. And so here, what I wanted to show in this picture, so it requires deep freezing of the samples, right, to keep their structure and properly hydrated. And so this is, a person in the laboratory of our own Tina Izzard here at Scripps, Florida, who is actually freezing a sample. And so one of the strategies that Scripps is using to stay at the forefront of research. That's only one of the multiple strategies they're using. But one of them is always to keep up with the latest technology and to be able to actually know what is the next technology that will bring the most advances in the science in the next year. So they determined, rightly so, that cryoelectron microscopy is one of those techniques. So they're going to acquire, I think Scripps is in the process of acquiring this machine here at Scripps, Florida. So that will make us the leading institution for molecular imaging in the Southeast. But I would say, even more importantly, it would help us solve the structure of P-alpha-synuclein star. And once we have solved that structure, we can design, the chemists and the molecular dynamics people are going to design little molecules that will be able to specifically bind and neutralize these little toxic aggregates. So we're working on that right now. Okay, so this was the first example of what we did to find out which protein clumps are toxic. The second example pertains to our understanding of how these clumps damage brain cells. So this is now another picture of neurons, and on the left you see good-looking, healthy neurons, so these ones are bipolar, and so they look healthy, at least to us. So we have model system where we'll have bacteria actually producing a protein for us in the right shape, and we show that it's not toxic, the neuron doesn't care. So this particular culture, we put on top of that the native form of a protein, and neurons are happy. If now we change the shape experimentally, and we add the clumps on these cells, they're not happy, they don't like it. This shows you the face of unhappy neurons. Can you see here how they're rounded up? Compared to these guys here. And also remember the garbage bags I told you earlier? It's full of them because all these little garbage bags, they are full, if we would label for them, we would see it. They're full of these protein clumps. They try desperately to get rid of them. So we wanted to understand, how they've come to this point. Why are they so sick? And then Dr. Minghai Chao in my lab, who's been working for many years on this project, what he discovered is this. So these are not the market trends of the New York Stock Exchange, luckily, because it would be really bad. What this is actually, what you're seeing here, is the decline of the molecules that carry energy in the cell over time once they've been exposed to the toxic protein clumps. They decline pretty dramatically in 4 days only. So you can imagine that's not really good for a neuron to have such an energetic crash. So we're working hard on understanding exactly the details of what happens, how this happens, what are the molecules involved that don't do their job so that the energy is lost like it is here. But the question we asked, keeping in mind that we want to develop therapies, was, is this even reversible? Is there anything we can do at this stage? And here comes the great news. It is. So we experimentally brought back in the neurons the molecules that are the energy carriers. And when we do that, even in the presence of the toxic proteins, the neurons revert back to normal and to becoming healthy. So it means that if you find a way to help the neurons combat these protein clumps, you will help them eliminating more or resisting to the aggression. So this is where it's really great to be at Scripps, because now when we find a point of intervention like we found in these two examples I showed you, we have the technology to start drug discovery campaigns and to bring this knowledge to be able to develop drugs, first molecules that will become drugs for the patients. So we did it for this project and for other projects. The first thing you have to do is to develop a test. A very, very simple test, a little reaction, if you want, that will fit in the wells of this 1536 well plate here. Why? Because this is what the robot will handle. And what you're seeing on the right is our high-throughput screening robot. It's a machine of extraordinary precision that is operated by Dr. Luis Campavia and Tim Spicer at the Drug Discovery Center here at Scripps, Florida. And when we have a test, we go to Dr. Campavia and Dr. Spicer, we show them the test, and we ask them to help us doing a drug screening campaign. And what we're doing here is that we're interrogating the chemical space. And we could do that at a speed of 100,000 different chemical molecules per hour, asking where is the molecule that will be able to do what we want it to do. In this case, restore the neuron's energy levels, right? So it worked wonderfully. And so now we're at the phase where we're moving the candidate compounds along the drug development path. What does that mean? It means that we're collaborating with a chemist here at Scripps. His name is Dr. Tom Bannister. And he changes little atoms here and there on the molecules. And then we test them. He gives them back to us. We test them in our biological system because we want to achieve the perfect drug, if possible. What is the perfect drug? It's a drug that is highly efficacious, very safe, you don't have to give too much of it, that is brain penetrant, we're working on neurological diseases, right? And that is stable, so you don't have to take it every hour or so, you can take it once a day, it's a pill, it's easy to take. And to move along the pipeline faster, together with Scripps, we spun off a company called VIT, which mission is to turn age-related neurodegenerative diseases into treatable disorders. So I have a lot of people to acknowledge. So first of all, I would like to acknowledge the people in my own lab. Dr. Minghai Chao, who was instrumental in the project I showed you about the neuroprotective approach. Dr. Diego Grassi, who did the work with, that led to the discovery of P-alpha-synuclein star. And Dr. Nicola Acera, Greg Ottenberg, Gianfranco Sferrazza, April Tobe, who is our administrative assistant and who does everything that I cannot do. She's amazing. And Dr. Charles Weissman, who is now Professor Emeritus at Scripps, Florida. He's the person who brought me here from France. And I'm forever grateful for him to bring me here, for the excellent, outstanding science we're able to do thanks to Scripps and the Scripps family. Our colleagues from the high-throughput screening core, Dr. Luis Campavia and Tim Spicer, chemistry, Dr. Tom Bannister. I told you about the role that he has now in helping us develop these drugs. Pharmacokinetics, Dr. Michael Cameron. He's the person who determines if the drug is going to go to the brain or not, if it's going to be stable enough. So it's really a collaboration now with all these groups together to push the drug development pipeline, and the animal behavior core led by Dr. Alicia Brantley. And finally, I would like to ask to thank you for being here and for your attention. Thank you.
Thanks, everyone, for coming. We're going to take some questions now. I have a microphone because I'm going to run around when you ask your questions, and we're video recording this today, so we want to get the question on the mic. The first question has come from our friends in the overflow classroom, so since they got the back seat, we're going to let them go first.
Okay, yeah, sure. I was reading it first to make sure. I'm slow. In your research, have you seen low blood pressure in Parkinson's disease patients? If so, is there a cause? So we're not medical doctors here. I mean, some of my colleagues are medical doctors, but we don't see patients. We're a research institution, right? So we don't have Parkinson's disease patients. However, it's a very well-known feature of Parkinson's disease, and it's called orthostatic hypotension. And this is due to the fact that the brain region which is affected, as I told you earlier, regulates movements of the body, but also regulates other functions of the body that are called autonomous functions that are also regulated by our central nervous system. I told you the brain is doing pretty much everything in our bodies, right? Including keeping the right pressure in our arterial and venous system so that we, stand and we're stable and we don't have these kind of symptoms.
And there's a second one.
Early on, diet and exercise recommendations to avoid the formation of these sick cells. Yes, so good diet and exercise, so it's two different things. Obesity is a very well-known risk factors for both Alzheimer's and Parkinson's disease. And this has something to do with the dysregulation of lipid metabolism and cholesterol metabolism. And there are a lot of studies now trying to understand at the level of the neuron itself, why is it so that if the lipid metabolism is not, is dysregulated, why does it make the neuron sick? We have some hints, but there's still a lot of research that's going on. But this is certainly for a fact. So this is why prevention of obesity is really a big part, an important part that we can all take to lower our risk of getting one of those diseases. Exercise, this is a fascinating field. And we have some colleagues at Script working on that. They exercise flies or mice. It's very interesting to see. You should come and visit. And so it was a black box for many years, but we start understanding more and more. And so what we know is that exercising does several things. It helps neurons regenerate. It helps neurons making more connections. It helps, having a healthy vascular system, so good blood flow in the brain, which the neurons also need. And it also helps making some molecules, some little hormone-like molecules that feed the neurons. So it does all of that. That's what exercising does. So there has been, for example, a clinical study in Parkinson's disease done recently where they found, so they had patients exercising a little bit, not exercising at all, and exercising a lot. And they found that really what makes a difference, at least for Parkinson's disease in this study, was exercising a lot. Sorry for the bad news, you have to exercise a lot.
So in your research, thank you for a most enlightening presentation. Very, very interesting. In your research, have you differentiated between dementia and Alzheimer's disease? Is there a different path for dementia?
So dementia is a group of symptoms. What dementia describes is severe impaired thinking, what we call cognitive skills. You know, the cognition is what our forebrain, the front part of our brains is doing. It's the thinking process. When that is very dysfunctional and people, you know, have loss of memory, can't think properly, can't make decisions anymore, that's dementia. So there are several diseases that cause dementia. Alzheimer's disease is one of the diseases that causes the symptoms of dementia. Dementia with Lewy-body is another disease that is molecularly related to Parkinson's disease. Fronto-temporal dementia is yet another disease that is actually molecularly related to ALS. Remember, ALS is the disease where the muscles become very weak. So there are different causes, and in each case, it's always a protein that makes clumps, but it's a different protein and a little bit in a different brain regions. That's why, you know, the initial symptoms that the patients have differ at the beginning, and then they unfortunately evolve into full-blown dementia.
Next question.
That's it. Thank you. I like that question. Yeah. Are donations accepted for speeding up research? Yes. And it does speed up research, believe it or not. Thank you for asking this question. I don't know who it was.
Notwithstanding the fact that imidacloprid and glyphosate and other herbicide and pesticides have basically poisoned our entire planet, especially in the United States. If you can at least put that over here, a lot of people are adopting the keto diet, which basically says no sugar, no high fructose corn syrup, low carbohydrates, a lot of fat, and moderate to fair protein. How do you see that diet affecting the causal nature of these kinds of diseases?
I don't think that there is any type of diet which will be causal in any of these diseases, right? My credo is that we should do everything in moderation. Remember, I'm of French origin, so we drink wine, we like to eat, etc. But when you don't overdo it, it's not going to harm your body. So all these diets, I mean, there is no, as I just said, causal relationship between any of them. As long as they're healthy and you don't overdo things, I really believe. I'm a little bit a skeptic of these very extreme diets. I'm unable to keep them myself. I think if people really want to try, they can, and you have to listen to your body. And not everybody's different. Some people, they do better when they eat more meat, like my husband. And some people do better if they eat a lot of carbs. That's what I need to do. So I think really the diet, it's every person maybe needs a little bit of different diet, and you have to try it out, and nothing too much in excess. The idea is, as long as you don't eat too much and you do enough exercise, you have a healthy body weight, and you know, a good energy level, that will lower your risk of getting any of those diseases.
Questions?
How close are you to find the treatment for the Alzheimer? And if 10% of the people who develop, women, Alzheimer, is there anything that they can do preventive medication-wise? They can take it before?
So preventing those diseases, that's the holy grail, right? That would be wonderful, that you don't even start having any symptoms. And there are studies that work on that and clinical trials that are done. It's called the Diane study. It's in families who have a familial form of Alzheimer's. So the families, so some of them are patients, but some are healthy people who just have a genetic mutation. They're followed up longitudinally. And then when they become sick, some of them become sick, they're given the treatments to see how they react to the treatments. And these are very good clinical trials because it's a homogeneous patient population, so it makes the clinical trials more effective, right? I think we have never been as close as we are now of developing disease-modifying treatments for these diseases. The advances in knowledge that I've witnessed, not only in my lab, generally as a scientific community has witnessed, for the last 10 years is amazing. all this, what I described to you about these proteins that get, the wrong shape, that make these aggregates, and that really kill the neurons, that's pretty recent knowledge. We knew that these proteins are around, but we didn't know what they're doing. Are they the cause, the consequence, etc. So now there is scientists are devising different type of ways to specifically target the pathways involved, to target these proteins, or even a risk factor, or a modulator, or a pathway, to be able, and that's really all what precision medicine is about. And unfortunately, why are we here now saying that there are only symptomatic treatments for these diseases is because for a long time it was not possible to do it. But now this has changed. I think we're not in an era of hope anymore. We're in an era where things are being done. And scientists are developing these treatments, so we need new clinical trials. Sometimes it's difficult to enroll the patients because there is a lot of reluctance due to previous failure. But, proper education of people who, like you, who come to learn about this disease, who understand how far we are and that we need everybody's help, including the patient's help, to participate, to be engaged, to ask us questions. I think the future is pretty bright.
Thank you again for your great talk. I have a quick question for you. You talked about how hard the brain works, and it's become quite faddish in Silicon Valley to do these dopamine fasts. So beyond sleep, they're thinking that if they could just relax their brain, whether it's through meditation or I don't know whether they're slack-jawed in front of a television, I don't know. But do you think there's any correlation between the overstimulation the brain gets in our industrialized society and maybe in other societies where things are more relaxed?
Yeah, so stress, excessive stress, actually any stress is excessive, right? We should never endure stress, is bad for the brain. So I think, what all these exercising and therapies are trying to do is to remove the stress from us. And certainly, I'm not aware of specific studies, and it would be very difficult to do. You would have to do a study over a long period of time, follow the people, and then make all the math to see if more or less people develop these diseases. But generally, stress in animal models specifically accelerates the development of these diseases, or sometimes it's the trigger for, the onset. So I think anything that removes stress is good. And then meditation or mental exercises are also good because learning is also something you can do to actually reduce your risk. Learning is very, very good. Why? Because when you learn anything, and it doesn't matter what you want to learn, whether it's something musical or a new language or coming here as you're doing today to learn new things, you're making more connections in your brain. And it's called the cognitive reserve. And the bigger your cognitive reserve, the more your brain is going to be resistant for these diseases. So any kind of these mental exercises, I think, is goes in the right direction.
I'm 72 years old. About 2 years ago, I totally lost my appetite, and my wife sent me to every doctor in the world. The question I have is the neurologist said I wound up with white matter disease. And I still don't know what that means. I was kind of told it's okay at my age. But I do notice a difference in my memory. And I was just curious if you could say something about that.
That's a difficult question for me because this is really not my field of specialty. I mean, I can tell you what the white matter is. The gray matter is constituted by, remember when you saw the picture of the neuron? There was the body in the middle, which kind of was rounded up, where you had the nucleus with the DNA, and then you have all these extensions, the arms that communicate with each other. So the body, where there are a lot of cell bodies, it's called the gray matter, right? And where there are the axons, the long projections, this is called the white matters. And some parts of the brain have a lot of these axons, these projections, so they look white under the microscope under certain conditions. But other than that, I'm not sure I really understand or I know what your doctor is talking about, and you would really have to ask him for more, Maybe you would have to change your diet to get more appetite.
Unfortunately, I'm told we're...
Thank you.